I want to thank you first for the invitation to present, and I'm very happy to be here today, and I will talk to you about 4-H. So I am from McGill University in Montreal, and then I will give you an introduction about 4-H and what we start, when we start, what we started, and where we're at right now. And then Dr. Wolf is going to talk about the key successes and challenges. So our work group was quite big. We were lucky to have people from different field, and I think we had a very nice discussion. So polarity-related leukodystrophy or polarity-related leukodystrophy is also, or polarity-related disorders is also known as 4-H leukodystrophy. 4-H standing for hypomyelination, hypodontia, and hypogonadotropic hypogonadism. So patients have hypomyelination, so they have neurological dysfunction. They typically would present in early childhood with motor delay or regression, and they'll have the phenotype is predominated by cerebellar features, so ataxia, dysmetria, dysarthria, and tremor. There are patients that present a little bit later with more cognitive and learning difficulties, and almost invariably patient will have pyramidal and extrapyramidal signs, most commonly dystonia, but usually it's not the predominant neurological involvement. We realized over the years that it's not only hypodontia, but multiple dental abnormalities, so oligodontia, delayed dentition, natal teeth. You can also have abnormal shapes of the teeth, etc. Same for the endocrine manifestation. So the classic is hypogonadotropic hypogonadism, which can present with delayed, arrested, or absent puberty. But we now know that about 50% of patients have short stature, and other hormone abnormalities are possible. I think I will let people sit down. It's okay. So the MRI characteristics are very specific for 4-H. So you have, I don't know, do you see my, oh, good. You have diffuse hypomyelination, and then you have preservation or relative preservation of myelination in certain structure. But you also have cerebellar atrophy in some patients, as well as a thin corpus callosum. So the structures that are typically preserved are the dentate nucleus, the optic radiation, the anterolateral nucleus of the thalamus, as well as the globus pallidus. So the genes for 4-H were discovered, the first genes in 2011. So it's a recessive disease, so it's caused by biallelic pathogenic variants in polar3A, polar3B, both found in 2011, polar1C 2015, and polar3K, if I'm not mistaken, 2018. The RNA polymerase III is one of 3 polymerases, and it's responsible for the transcription of small non-coding RNAs. We're not going to go through all this, but that's a nice summary of all the small non-coding RNA that are transcribed by POL3, and they're all very important for cellular homeostasis and for translation. So as all of you probably know already, when a gene is discovered, then the spectrum enlarges. So we realized that if you have a specific combination of polar3A variants with a leaky splice at a very specific position in polar3A with a stop mutation on the other allele, you have a very severe disease with an onset between 1 to 3 months of age, with severe developmental delay, regression, severe dysphagia, failure to thrive, and earlier death. And there's a specific MRI pattern with this form of the disease. And then for the typical patient that we talked about, typical onset in toddlers and some later childhood presentation, and again, a typical MRI finding. And then we also know now that there are adult onset of polar3-related leukodystrophy, with typically more myelin in the brain. And also we do know that some of these patients are diagnosed incidentally because they have an MRI for another reason. So they probably get a much later disease, but they're probably very under-recognized. But then the disease spectrum expanded even further when we realized that hypomyelination was not obligate for 4-H. So that's where the Polar 3 related disorder names started to be more used. And then we also realized that you can have ataxia and specific mutation in Polar 3A. You can also have a hereditary spastic paraparesis type phenotype as well. And then we also realized that some patients have a progyric syndrome with specific Polar 3A mutation. And then when we found the gene in 2015 for Polar 1C related leukodystrophy, we realized that there was an overlap with Treacher-Collins syndrome. So some patients have Polar 1C mutations and Treacher-Collins only. Others have a leukodystrophy only, but some patients have both. And finally, very recently, we realized that some very specific sporadic dominant mutation, de novo mutation in Polar 3B leads to a completely different phenotype of ataxia, spasticity, demyelinating neuropathy, and for some patients, epilepsy. So the research challenges and successes. So I think one of the huge success of the last few years is the patient advocacy group, the GLIA Foundation for H leukodystrophy, who received a RARAS-1 grant from the Jan Zuckerberg Foundation and has started a registry for the patients. One other success is the understanding better the molecular disease mechanism and some development of disease models, some of which are published, others are not. Some of the challenges that Dr. Wolf will discuss later is, of course, for future clinical trial is the clinical heterogeneity, as well as variable cellular targets and the lack of biomarkers. And at this point in time, there's no clinical trial for this disease. So a little bit of background about the successes. So we now know that, we know it's a hypomorphic disease. So you can have the disease if you have less amount of a specific subunit. You can also have some variants that lead to abnormal assembly of the complex because of abnormal interaction between two subunits. And you can also have disease because of decreased interaction with the DNA for transcription. So there has been several transcriptomic studies. Unfortunately, it's hard to have like a clear pathogenesis mechanism. The tRNA seems to be a very important driving mechanism, but the studies have been done on different form of the disease with different cells and different phenotypes and different experimental approaches. So it's hard to conclude at this point, but hopefully in the next a few years, we'll be able to have more data on the RNA-seq and understanding better the transcriptomic aspect of the disease. So there's been some published and unpublished animal and cellular models. So I'm going to briefly talk about the mouse models. So we know that if you have a homozygous knockout, it's embryonic lethal. There is the group of Bernard Bray who did a Polar 3A with a common Quebec mutation for Polar 3A. The mouse has no phenotype. Like another Polar 3B mutation was tried, but the mutation causes too severe of a complex assembly defect, so it's embryonic lethal. And then another thing that they tried is to combine homozygous Polar 3A mutation with one copy of the severe Polar 3B mutation, and again, no phenotype. But recently, Dr. Willis's lab published a mouse model of Polar 3A with two subsequent Polar 3A mutation. This combination of mutation was chosen based on yeast studies. And this is the first animal models of the disease, because that's very good news for the community. The mouse has a growth phenotype. It has a neurobehavioral abnormalities. Unfortunately, fortunately, fortunately impaired myelination, but unfortunately no motor deficits. And no non-neurological features, but still a very good model to understand the disease pathogenesis and hopefully eventually test some potential therapies. So clinical challenges and successes now. Clinical care is well described, including the systemic manifestation. We're still working on them, but we've done quite a bit of work regarding this. The diagnostic, I think we've heard it very well in the PMD session, is dependent on the availability of next gene sequencing, either genome, exome, or panels. So it's not every countries that have access to this. There's no pathway to newborn screening at this point, and I think the diagnostic awareness is still a challenge. There's still some, the disease is not as well known as other leukodystrophies. I think the clinical care has been relatively well described, but still there is work to do to understand, for example, the benefit of hormone replacement therapy for patients with hormone deficiencies and optimize versus bone health versus potential side effects. Same for the ophthalmological manifestation, which is most commonly myopia, and it progresses over several years, so it's important that the patients are followed regularly by an ophthalmologist, and it's often become very severe. So, and lastly, there's no disease-modifying therapy available, but hopefully at the next or a couple of meetings later, we'll have good ideas for this disease. So the Heatmaps, so there's some red and orange, but more yellow than what we used to have. And I think for a relatively new disease, it's encouraging. And hopefully next time we'll have more yellow and perhaps some blue. I'll let Nicole Wolf talk about the challenges and successes.
Thank you, Geneviève. I have to say that I'm quite glad that this presentation is after the Pelizaeus-Merzbacher presentation, because both leukodystrophies are hypomyelinating leukodystrophies. But I personally have much more trouble understanding the disease mechanisms in 4H than Geneviève just explained. It's as she said, it's a very new disease. I mean, the genetic cause was identified by Genevieve's lab in 2011, so that's not very long ago. So we have a long way to go until we really understand it. And it's not a simple enzyme like arylsulfatase A in metachromatic leukodystrophy, where we know if the enzyme is not there, you have this build-up of sulfatides. Here it's much more complicated. I also, perhaps it's not right, but I also always think that the polar 3 role is so tightly regulated because it's such a central process in a cell working on transcription that probably we have much less leeway to influence that activity, which makes it quite a difficult disease to treat. I think the biggest success, at least in my eyes, is the nice collaboration with the Montreal team and also the team of Adeline. This transatlantic cooperation has led to large studies in spite of actually scarce funding. I think I could say that. And that's also thanks to a lot of you who contributed patients and didn't write a case report, but contributed your patients to those large disease series. So all three of us are very grateful for this. I think that's my successes. I go on to the challenges. We have still difficulties to understand the genotype-phenotype relationships. I come to some of the specific variants which give you specific disease forms in a minute. Geneviève has already alluded to it, but it's still not well understood. The disease models you just heard, they are coming. There are some mouse models underway, and we hope that they are good replications of the human disease or parts of the human disease. And the other question I have is 4H leukodystrophy a leukodystrophy at all? And I will show you an example. Of course, I think it is. But we see so many patients now without leukodystrophy that I think we have to ask us this question. And that actually also entails the problem, how relevant is the myelin deficit in this disease. So Geneviève already told you about the different variants, and all started with the 4H leukodystrophy, the typical MRI here, and we also have severe cases, milder cases, cases with a cognitive presentation. But we also have patients without hypomyelination, mainly with variants in polar 3b. We then have the severe Wiedemann-Rautenstrauch syndrome caused by specific variants, severe loss of function variants in polar 3a. We have the polar 3a related basal ganglia disease, and all patients have on at least one allele, one of those splice site variants. And then we have the group of patients, I have never seen one, with a later, often adult onset spastic ataxia, and on one allele, this intronic variant in polar 3a. And how all those disorders are related and what causes one or the other is not well understood. There has been a big progress, of course, in the structure of the human polymerase III. There is now a crystal model. In the first publications, all the variants were mapped on, I think, a yeast polymerase III. So that helps already, of course. You see that some variants seem to cluster in some parts of the protein. Especially the Treacher-Collins variants caused by polar 1c variants. You have this varicella zoster virus susceptibility, which gives you a very severe varicella zoster infection. That's quite interesting because the very first 4H patient in my very first cohort I saw in 2020, in 2000, so more than 20 years ago, had already had indeed quite a severe varicella infection. And we do have patients who have an immunodeficiency, and that is something completely, not researched at the moment. So we start to have an idea where some mutations are located in the protein and give you the specific forms, but why it's still a problem. So what is the primarily affected cell type? Is it only one cell type for the typical forms? Is it oligodendrocytes because of the hypomyelination that seems to be obvious, or is it neurons? And if it's neurons, is it cerebellar neurons, cortical neurons, or the deep gray matter neurons in the basal ganglia variant? And what of the polar 3 functions is primarily affected? Is there different between the very different phenotypes, and how tightly regulated is it? And what I mentioned already, how careful do we need to be in influencing it? I think these are all important questions before we start thinking of treatment trials. So what we showed some years ago, that in the patients with hypomyelination, so the typical MRI, the severity of the myelin deficit correlates with the clinical severity when you map it like that. It's not a very strong correlation, but it seems that was our idea. And I think when I think of my hypomyelinating 4H patients, I would also say it confirms what we see in the clinics. We've seen recently some very severely affected typical 4H patients, and they have a very hypomyelinated brainstem. I don't know whether you have the same experience, and that's perhaps something we need to look more closely at. So that seems to be quite straightforward. It's also the case when you look at the patients with Pelletier-Merzbacher disease and the degree of myelin deficit, there you also have a correlation between the severity of myelin deficit and severity of clinical presentation. And perhaps also important to remember that there are patients with quite severe hypomyelination and actually almost normal motor function. This is something which surprised us, of course. And when I was seeing those first patients as a registrar in child neurology, I actually went to Odile Bösflug-Tanguy, who is an expert on Pelletier-Merzbacher disease because it's a hypomyelinating disorder. And I asked her, have you seen this kind of patients? And she actually didn't believe that those 4H patients were able to walk. She said that's not possible with an MRI like that. So that's how it all started. So of course, those patients, we all know them. There are many patients who are quite mildly affected. And those are two patients I see in my clinics, both of them with Polar 3B variants. And when you've seen many patients with 4H, you recognize them. They have this severe myopia. They have a very typical behavior also. Many of them are very friendly. Some of them have quite some autistic features, which is also an under-recognized part of the presentation. But without being dysmorphic, their facial features are very much similar. So, and they are ataxic, so these patients look quite alike, but are still different. And I would like to ask you a question about clinical severity. So this is one patient you see with a normal myelination and normal corpus callosum, cerebellar atrophy, and that patient has a clear hypomyelination, a thin corpus callosum, cerebellar atrophy. Both are 18, 20 years old at the time of the MRI. And who of you thinks both of them are similar in their neurological severity? No, not. Who of you thinks this one is more severe? This one, the hypomyelinating one here on the. This one.
Yes, but I'm guessing you're going to say no.
Okay, so this is a patient who is 20 years old. He has a normal IQ. He has a mild autism. He has a mild ataxia, which is very slowly progressive. He has myopia and delayed puberty. That girl is 19 years old. She has moderate intellectual disability, a clear ataxia. She's barely able to walk without support. She also has myopia and delayed puberty. So that's my problem. And that's, I think, one of the challenges in 4H, where we have to ask ourselves what is the most important part in this disease to try to influence, for example, in a clinical trial. Is it the myelin? those two patients would perhaps argue no. Is it the cerebellar atrophy? Then we have a problem, because how could we influence, how could we treat that? So I really am very curious as to what you think about that question. And I think probably you have made the same experiences in your patients.
Some patients don't have cerebellar atrophy,
Yeah.
but very, very ataxic.
Yes, that's true as well. So Geneviève just said some patients don't have cerebellar atrophy, but still are very ataxic. So that makes it even more complicated. Yeah. The other thing is the primary outcomes in clinical trials. I think because we have those really different presentations, like the basal ganglia phenotype or the spastic ataxia with those specific genetic variants, that we need to stratify clinical trials alongside those important presentations. The question is, do we need to look at stabilization or improvement? I think that's a question for all of the leukodystrophies, and stabilization would be the first goal. But it's a very slow disease in many patients. To look for, certainly in those very typical for age patients, to look for stabilization is something which is also, given the scarce, precise natural history data we have, still a challenge. We don't have established body fluid biomarkers. We don't have good biomarkers for myelin. For example, what Mark Engelin yesterday already said, the neurofilament as the universal neurology marker is also not well understood, although I think that it might be helpful, but we need many more patients to really understand its role for 4H syndrome. And I think we have also to look for better biomarkers. The question is what? Other open questions I have just from seeing quite a few 4H patients. We have, of course, we have this Wiedemann-Rautenstrauch syndrome, which is a progeria syndrome. But also in typical 4H patients, we see certainly as they get older, progeria-like signs in those patients. We have the immune deficiency in some patients, and I think as soon as you start really looking for example, antibody production after vaccinations, we probably find many more children who have those problems. And then the failure to thrive and growth deficit, half of the patients have. Does it correlate to the severity of the neurological involvement? I would think at least a part of it. So those were my thoughts, and I think we are both grateful to the support we had to look further in this difficult disease. Thank you.
So we have some time for questions.
Thank you. This is great. What about other features of the MRI? So all the diffusion-weighted fiber density, and all kinds of other things that I'm sure spectroscopy, et cetera, et cetera. Did you look at those things to see if you can explain some of the surprising findings that you described?
We did look at, but it doesn't help us really further at the moment, at least.
Great talks. I was curious, and this just shows my ignorance here. What is known about the early developmental expression of Polar 3A, Polar 3B, and wondering how much of this is a dynamic that is unfolding postnatally that is really reflecting something that's happening embryonically and that we just can't put our finger on because we're coming late in a developmental program.
That's a very difficult question. I don't think we have an answer to that. We know we have patients that have a congenital form of the disease that we were recently aware of. I think that if you want to a general hypothesis is that there's a threshold for you need transcription of these very, very important transcript. And so if you don't have that threshold, you have disease, but I think you probably have different types of threshold according to how severe the disease is and how much more you get from on top of the hypomyelination. And also we're looking at that more deeply, but looking at specific variants, the 1771 -7 and -6 are very, very specific for basal ganglia involvement. And we're also trying to find out exactly what happened. I think it probably starts in the putamen and then you get in degeneration and involvement of other structures afterwards. But that's, and I wonder whether or not the Polar 3B patients that don't have hypomyelination also have a very different mechanism. We do think that the de novo Polar 3B variants leading to developmental delay ataxia and peripheral neuropathy and epilepsy also have their own mechanisms. So I think it's going to be, hopefully we'll be able to have different mechanism for different form or mutation.
Yeah, I agree. Why are, for example, in the basal ganglia phenotype, the basal ganglia are affected, and what does it mean? Also, when you, what does it mean for Polar 3 function? What does it mean when you try to design treatments? Because it's a leaky splice site, so there is some function, but how exactly is it affected to lead to this putaminal abnormalities? So that's puzzling, at least for me. I also think that the non-hypomyelinating Polar 3B patients probably have a different disease mechanism. As a group, I would say they have a more severe intellectual disability than the typical 4H patients, they have, if patients have epilepsy with the Polar 3 variants, it's usually the normally myelinated patients. So I wonder whether that is a cortical neuronal phenotype in contrast to the basal ganglia form. But this is, of course, all hypothesis. Those patients, although you can perhaps even separate them from the classical hypomyelinating patients, still look like the same disease, if you understand what I mean. The basal ganglia patients look different, but the hypomyelinating and the non-hypomyelinating patients, if I would have 6 of them at the same time in my waiting room, I think everyone would say they have exactly the same thing. And also interesting that in the last 3 or 4 years, we identified more of the non-hypomyelinating patients due to exome sequencing than the typical 4H leukodystrophy patients. I don't know whether you had the same experience.
Just related to that, is there any correlation between the remnant? Have people looked at the remnant enzyme activity of the polymerase and whether that correlates, the enzyme activity correlates with the severity of the disease or the phenotype?
So it's it's not an easy assay to do. And if I can ask Dr. Willis to please comment on that, do you mind?
Oh, she's coming.
The correlation between structure and function, really, we don't have that yet. The assays are involved, but they're certainly very doable, and it's just a matter of getting the mutations made in cell lines, purifying the enzyme and doing the work. So it's a very feasible thing to contemplate, is trying to correlate disease severity with mutational severity and enzyme activity, and I think this is a worthwhile endeavor.
But there's more than 250 mutations, though.
But the assumption is it's not a null mutation, right? That there is the assumption is that there is remnant activity.
You have to have.
You have to have
some
polymerase.
activity. Yeah.
Yeah, but is it only the enzyme function, or are the other functions also important, and in what way?
Question.
The basal ganglia phenotype, we've got a couple of patients with abnormal DAT scans, surprisingly. And one of the questions is whether we treat them with L-DOPA. I haven't done that yet. But do you see that, and have you tried that?
I treated one patient with L-DOPA, but it was a patient that was living further away, so I couldn't have my I couldn't examine him in person as much as I would have liked to. But he was not very affected. So the patients that have they're almost, I guess, for 1771 -7 or -6, or they have one of each, they're relatively mild, at least in my experience, and the progression of the disease is very slow. So the dystonia is not as problematic that you want to treat them, but I would do for any young patient with dystonia. I usually start with Cinemet and see.
Perhaps I may add that we saw also quite a few patients with the basal ganglia phenotype in the last years. Again, more than the high leukodystrophy phenotype. There are those very severely affected young children with infantile onset, and there are those more mildly affected patients with an onset in early childhood and a slowly progressive disease, which most looks like juvenile Parkinsonism. And we did treat various patients with L-DOPA without any success. And very frustrating in those patients is the bulbar involvement. They develop anarthria and severe dysphagia. And there's also the possibility of a huge intrafamilial variation of disease severity also in this group of patients. I have a brother and a sister, and the sister is almost not affected besides a mild learning difficulty, and her brother is almost not able to speak anymore and has a severe Parkinsonism. And the same MRI, by the way.
Yeah, and the infantile form with the patients that have a stop in a -7 or -6 is also very different. The MRI pattern is a combination of some hypomyelination with basal ganglia involvement and cerebral atrophy, and their movement disorder is different. They're very hyperkinetic. They're very dyskinetic. So they're also recognizable in the waiting room.
And you wouldn't do a DBS for that anarthria or the dystonia? It doesn't get that bad.
Well, we discussed it with the DBS colleagues, and I saw two patients with the DBS team. And just the anarthria is something which they think will respond not well to the DBS. Would your...
Right. Oh.
I find it does help some patients, anarthria as well, DBS, as well as the dystonia. I can't tell you why, but...
They have a lot of oropharyngeal and
Yeah.
Well, thank you very much. I guess if there's more questions, we can do it during the break. Yeah, on it.